We're starting our third unit with blood, and let's take a look at this chapter. So we're going to be talking about the components of blood. There will be the plasma, which is the liquid portion of blood, and then the cellular elements of blood, so that would be the solid portion of blood. We're going to also take a look at blood cell production, and then in particular red blood cells and their function, and then also platelets and coagulation. Okay, so first, the components of blood. So we have blood as a whole is composed of the liquid portion, which is called plasma, and then the solid portion, which would be the cells. But first, here on the plasma, the vast majority of plasma is water. And then we also have electrolytes in there, ions, as you know, sodium, chloride, other ones, bicarbonate, for example. And we have organic molecules in there, and we're going to get back to these in just a sec. And then also trace elements and vitamins in the plasma, and gases such as CO2 and oxygen, dissolved, not bound to hemoglobin. We're talking about just dissolved gases in the plasma. Now, the organic molecules, we're going to have amino acids, proteins, glucose, lipids, nitrogenous waste products. Among these, of course, the amino acids and glucose and lipids, those are important nutrients, and then here's some waste products. But let's take a look at the protein component. And there, very importantly, we have albumins. And in the lab portion that goes with the blood, I told you already that the albumins, that's the most important factor for the colloid osmotic pressure. And that is very important in order to recapture fluids at the end of capillary beds. We also have globulins such as immunoglobulins like IgGs, those are the antibodies. And then also an important protein is fibrinogen. That's the precursor to fibrin, which is very important for blood clotting. So here, the function of the plasma proteins, the albumins, very important. It's a generic kind of housekeeping protein made by the liver, but the big thing here is that it's the major contributor to the colloid osmotic pressure. And I really want you to understand this concept of the colloid osmotic pressure. Also, you can use albumins to carry various substances that are lipid in nature, for example, steroid hormones or thyroid hormone. And then we have globulins, and they're important for mostly it's antibodies, and then we have some clotting factors, enzymes in there. Fibrinogen, they are made by the liver, and they're very important for blood clotting, very important. So fibrinogen is the precursor to fibrin for blood clotting. And then we also have transferrin, that's important for iron transport. Now here we are taking a look at the solid portion of the blood. These are called cellular elements, and the vast majority of these cells in blood are these little red blood cells. They look like a little donut with relatively small cells, about 7 to 8 micrometers in diameter, and they of course are filled up with hemoglobin, and the hemoglobin then carries the oxygen. Let's skip down first on the platelets. These are little chipped off pieces from megakaryocytes, and we'll take a look at them later. But so they're not considered cells, they're just fragments of cells that chipped off or shaved off of these large megakaryocytes. Now, your immune functions, they rest with these white blood cells. And if you listen to the recordings on the lab, you would already know that we have granulocytes and agranulocytes. So the granulocytes are from here down. There will be neutrophils, eosinophils, and basophils. And then on the agranulocytes, we have the lymphocyte and the monocyte. And so let's first start here with the granulocytes. By far, the most numerous white blood cell that we have is the neutrophil. And it's called a polymorphonuclear neutrophil because it has these many shapes on the nucleus. Basically, it looks like somebody punched the nucleus and it just kind of broke into pieces or shattered. And so 3 to 5 lobes, and we talked about it in the lab. The neutrophils, they have granules. That's why they're in the category of the granulocytes, but you can't see the granules because we usually don't offer the neutrophils a neutral dye that would stain the net granules, but the nucleus is so obvious you can't miss the neutrophils. Now, the eosinophils and the basophils are very similar. They're full of little granules, and the only way to distinguish these two types of cells would be to stain the granules. Now, the eosinophils, they like eosin as a dye, which is an orange dye, and so their granules will show up in this orange kind of color. And then the basophils, well, they like the basic dye, that's the crystal violet that we offer them, and then their granules will stain purple and pretty dark cells. These both are very rare. Eosinophils are about 1 to 2% of your white blood cells in circulation, and these basophils are even more rare because they turn into mast cells once they migrate into tissues, which they pretty much do right away. Now, going back up here to these guys, lymphocytes, that's your third line of defense. Those are your most important immune functions, very specific immune functions. These guys, they will differentiate into B cells and T cells. And we'll get to that in more detail in that immune system lecture. The monocytes, you have about 5 to 7% of your circulating white blood cells. Our monocytes, they are the largest of these white blood cells. They have kind of a kidney-shaped nucleus. And once they migrate into tissues, then we call them macrophages because they're big eaters. They eat up debris, macrophages. Big eaters. Okay. So here, the write-up for these cellular elements. So we have the vast majority being red blood cells. Then we have the tiniest ones. Those are platelets. Those are shavings from the surface of megakaryocytes. And then the white blood cells, again, we have the lymphocytes and the monocytes. These two here would be agranulocytes. And then the granulocytes are these three types of cells. So here you have these granulocytes. And these are agranulocytes, these two, lymphocytes and monocytes. Now, we're going to take a look at blood cell formation. The mother of all of these blood cells, whether red or white blood cells, or even the platelets, megakaryocytes, the mother of them all is called hematopoietic stem cell. Hematopoietic stem cell. That one is in bone marrow, and it basically can generate all of these different white and red blood cell lines. Now, the way it works is the hematopoietic stem cell, when it divides, it regenerates another stem cell, and then it will also have one cell. The second cell is then going to differentiate. And so we have this uncommitted stem cell at first, and then these can make all the different cell lines. So here, if we go over here, then we would make eventually red blood cells. Over here, we would make megakaryocytes, and here all of these different white blood cells, except the lymphocytes, they have sort of their own special pathway right here. But nevertheless, the hematopoietic stem cell is the mother of them all. So here, let's take a look at some of the end products here of once the hematopoietic stem cell has made these white or red blood cells. These are the differentiated cells that are in circulation in your blood. The immediate precursor to an erythrocyte, a red blood cell, is called a reticulocyte. The reticulocyte still has a nucleus, and so therefore it doesn't have this sucked-in center that makes it look like a donut. It looks more like a regular cell. Once it gets rid of its nucleus, then it has this donut shape of a typical red blood cell. Here the platelets, again, these are little chipped off fragments from these megakaryocytes, and then your 5 different types of white blood cells. Okay, so let's take a look at hematopoiesis, and we're going to first take a look at the hormones that are necessary to drive hematopoiesis. So first one here would be EPO, erythropoietin. That's a hormone that's made by the kidneys. It's released by the kidneys in response to low oxygen content in the air. So if you go into high altitude, then the kidneys will respond to this lowered oxygen levels in the air with releasing erythropoietin, and that makes more red blood cells. I think I mentioned this also in the lab. Erythropoietin is pretty popular among athletes because, more red blood cells mean more oxygen, means more endurance. If you do it the natural way, if you go up to altitude camp and you expose yourself to a lower oxygen content in the air, then it's legal, completely legal. You can do this. You will generate more red blood cells, and once you come back down to sea level, you will have more endurance. Now, injecting erythropoietin without going to altitude camp, that's not legal. That's considered blood doping. And then here, another cytokine that's involved in hematopoiesis would be the thrombopoietin, which then leads to the formation of more megakaryocytes, and that will be what we need to make more platelets. And then we have colony-stimulating factors, interleukins, and stem cell factors. There's a bunch of them. The CSFs, the colony-stimulating factors, there's a whole host of them, and the interleukins, they're very numerous. There's a lot of communication going on between these white blood cells, but we will touch on that more in the immune system lecture. Okay, so here let's continue with in general the hematopoiesis. When you're making white blood cells, that's called leukopoiesis, and when you're making red blood cells, that's known as erythropoiesis. The hematocrit is an important number because it specifies the percent of your blood that is solid. So if you take a small amount of blood and you put it into a capillary and then you centrifuge that capillary, then by percent, the solid portion is the cells that settle on the bottom in your centrifuge tube, that is your hematocrit percent. And here's what that could look like. Here's this capillary, and we would have done this in the lab, of course. Here's your whole capillary top to bottom. That would be 100% up to here. So here's your 100% line. And here in this case, we have 42% of the length of this capillary tube is packed cells, plus a little bit here of a spuffy coat, the white blood cells are settling on top. But this here is the solid portion of the blood, and so this is 42% for the hematocrit. And then the remainder, the liquid portion of the blood, the plasma, is here 58%. But the hematocrit measures the percent solid of your blood. And so here, we saw this already in the lab. Males typically have higher hematocrit, higher hemoglobin levels as compared to females. So you can tell there's a little bit of overlap, of course, but in general, males have a significantly higher hematocrit and hemoglobin level, and that is due to the higher muscle mass that males generally have. The red blood cell count should be about, if you don't consider gender, it would be about 5 million per microliter of blood. And again, for females, a little bit lower as compared to the male numbers, but the ranges are fairly big and there's some overlap. Total white blood cell count should be, here they're putting it 4 to 11,000 per microliter. That's about right. If you see somebody with a 15 to 20,000 microliters of, let's start over. If you have somebody that has 15 to 20,000 or so white blood cells per microliter, that will be considered probably this person is going through an infection, so slightly elevated, would not be raising much of a concern. But if you're going past 50,000 and then approaching 100,000, that could very well be a leukemia. Okay. The differential white cells, here by percent, you're supposed to have the vast majority of your white blood cells should be neutrophils. And so here you have 50 to 70% of your white blood cells should be neutrophils, 1 to 4% eosinophils, less than 1% basophils. Right here, and then we have lymphocytes, about 20 to 40%. For your B cells and T cells together, and monocytes approximately 2 to 8%. So those are roughly the numbers that we had also in the lab. And platelets, those are these little chipped off pieces from megakaryocytes. You're supposed to have quite a few of them. You need them for blood clotting, and if you're willing to donate platelets, all hospitals are happy if you were to do that, because platelets, they only last for about a week, and if somebody lacks platelets, they are relying on those donations, and so you do somebody a big favor, if not saving their life, with a platelet donation. So here we're going to take a look at the stem cell and the blood cell formation sort of in pieces in the bone marrow. Here you would have a stem cell. We're going to take a look at that here. A few various white blood cells that are shown here.